and these incredible updates going by welcoming to the stage two people who are just incredibly close to our hearts, our dear friends, incredible researchers and innovators from Murdoch in Australia, Simran Kaur and John Christodoulou. Thank you guys so much for joining us and talking about all of your work that's happening. Thanks, John and Sim. Welcome.
Hi, Luke. Pleasure.
Hi, Luke. Hi, everyone.
Hi, Sim. You guys are so kind for joining us. We really appreciate it. And I know the Sim, your presentation, just like all of your communication that you're constantly bringing to us, means the world to us. So thanks. And I think best maybe just to dive into the questions, and I'll stop talking. So we have a couple of questions for the team at MCRI that you guys we're hoping you can speak to. And the first one is one that that comes up a lot, but we're so excited when we hear these updates about where you are in the process in the high throughput screen and search for small molecule drugs to hopefully treat KAND, and what are some of the potential next steps.
So probably I can start a little bit, and John can add some of his viewpoints as well. So first of all, I think for any high throughput screening, we do need a great team, a great collaboration, a method that works, and a method that is shown in the lab to work as well, which is optimized. So I think we have got to this. We are stepping up the ladder pretty quickly, and we've got all these resources in hand, which is great. So far we have worked up the workflow, we have optimized it, and we have done some pilot screens as well. We've got some data, but probably it is too early to say anything too specific about that. We need a lot of testing done to be very confident about the results that we are getting. But I would like to just add in one point here that in the high throughput screening, while we are testing the other drugs, we do want to focus on those drugs as well, which is FDA approved, which have been previously reported, or they are shown to have an effect on the related neurological conditions. And COVID has been a bit of a play factor here, because usually these drugs have to be imported from somewhere, and it took us about 3 or 4 months getting those drugs. So we are creating a list of drugs that we want to focus on, and we are just getting them shipped as quickly as possible. But this is what it is at the moment. But yeah. And I think some of the next steps that we want to do is like, it's interesting to listen in the previous talks as well, that if you want to do any modifications in your assay to make it even better, do it quickly and do it sooner. So this is one thing we are working at the same time, just with the pilot screens that we have run. There are a few things that we want to change in the assay, and our collaborators, they have been really great and super helpful to make suggestions about what we could do to make it more reliable, more efficient, so that the similar approach could be more widened into the variants that affects the KIFani. So yeah, so this is what we are at the moment.
Thanks, Sim. You mentioned in your presentation, which again, I'll say, I hope everybody goes back and watches 3, 4, 5, 6 more times, because I learn a lot every time I hear from you all, which is often. Thank you very much. You mentioned in your presentation that 100 molecules have been identified to be synthesized and tested in your lab. How long does it take? What's the process to test 100 identified molecules? That seems like a really rigorous and lengthy process, and you guys have been working so hard. I'm wondering if you could speak to that a little bit.
So with the 100 molecules, so the team at AMRI has been a great collaborator. So this is what they have identified using the computer-assisted drug design methods. Now these 100 molecules, it shows the backbone structure of a molecule. It could be that little tweaks could be made to these molecules to make them even like a library of more than 100, probably 300 or 400. But just focusing on these 100 molecules, what I would say that the most time-consuming part, which is going to happen, is the design of these molecules, because most of them, they might not be commercially available. So we have to find some manufacturers or some companies that could custom design these molecules for us. Now this process, with my understanding, could take a bit of time, and maybe it could be from 3 to 4 months. So this is something that is going to be the very time-consuming part. And after the drugs have been designed, the next step, which is the high throughput screening-based method, it's not going to take much time. Probably it'll just take about 1/3 of the plate, and it could be processed in batches with other molecules that we are testing. So yeah, the time-consuming part is the manufacturing of these drugs.
Got it. Right.
And,
John, yeah.
and maybe I can add, Luke, parallel to that, we're also screening a bunch of already known drugs that have been FDA approved and that are in clinical practice. So you can get these drug libraries off the shelf. So 2,000 or 4,000 drugs that you can then test very rapidly using the high throughput screening system that Simran's developed. And in that way, we can, if we're super lucky, we'll identify a drug that's been used for some other disease for years, has a good safety profile, that's approved by the FDA, that if we validate it in the high throughput system using iPS cells as the next step, then very rapidly we could jump from that straight into clinical trials, and may not even need to have to worry about going down the path of animal models, for instance.
That's great. That's really exciting and inspiring to think about. And I want, another thing, Sim, that you mentioned in your, and John, we've talked about it before too, in your presentation, is the similarity between other kinesins. And I'm wondering if you might touch just a little bit about how identifying similarities in KIF1A affects maybe identifying possibilities for other kinesin-based diseases, but as it applies to KIF1A, what understanding features and characteristics of other kinesins, how that applies to, could apply to supporting KIF1A therapeutics.
So with the so now we know that the kinesins, they have got their the motor domain, which is a part which we call as a head part of the protein that walks along the microtubules in a very ATP dependent manner. Now, when we look at the conservation throughout the sequence, there are sections within the kinesins which are really highly conserved. And one of that part is motor domain. It is very highly conserved in the kinesin superfamily. Now, this could be a great resource that we could harness, like if there is any other research, especially about the small molecules which is going on in the field, not specific to KIF1A, but other kinesins, for example, the kinesin 5A, B, and C, that we could harness some information from there as well. So in the again, small molecule research in kinesin overall field is not very, it's not at a very super fast level. So, but there are few published research that shows some of the molecules that have been shown to have an effect on the positive effect on the movement of KIF5B motors in the high throughput screening manner using the different assays. So probably this is what we have touched in the presentation as well, that we could take those molecules and test if they have any effect on the KIF1A, thinking that because the motor domain is so conserved, it is showing the similar mechanism, the similar change in mechanism of the kinesin. So whether we can see if it has got any effect on KIF1A at all or not. So this is, I think, this information we could use just to see if it works on the KIF1A or not.
Thanks, Sam. Yeah, and another question that came in is how do you look at a subset of mutations versus a single mutation versus cluster, and will the drugs identified in the screening possibly be able to affect all KIF1A mutations, or focusing on subsets of those mutations along the gene?
Mm-hmm. So I think our approach here is just to look at the mechanism which is resulting in the loss of KIF1A function. Now, with I think the reports from our research roundtable network and all the publications that we have reported so far, it shows that although there is a clinical heterogeneity, there is it's quite different that each variant in the KFANE affects its function in a different way. But overall, if we think about, there are possibly two mechanisms which is of great concern to us, especially in the motor domain variants. First is the ability of the motor protein to attach to the macrotubules, and the second is its ability to move at a pace which is required for the effective brain function. So I think if we think about these two broad mechanisms, we could definitely apply a small molecule which is affecting, let's say if a variant is affecting the speed of the motor, and we have shown a small molecule which helps in increasing that speed. So probably the other variants in that pocket, in that nearby regions, which is affecting the same mechanism, the small molecules could then be widely tested for all those variants to see if it has got an effect or not. So it's like kind of a banded approach for the variants that's showing the similar effect on the function of KFANE, and testing if the small molecules could correct that.
That's really, that clarifies a lot for, I think, a lot of the questions that have come in. So thank you, Simran and John. Thanks. If there's, I feel like I could, like many of these the conversations that we've had, I could go on and on forever. And there's certainly the questions that keep coming in, and I'm sure we'll be sending you off pages and pages of questions. But it's remarkable to me how just incredible over the last, however many, almost 2 years that we've known each other, how collaborative you are, and how you guys are really just working and partnering with so many different people to get to the top of this mountain as fast as possible. And can't thank you enough for that. Truly, it's so apparent, in our entire community recognizes that, and all of the work you do means the world to us. So I would say, Sim and John, if there's anything, any message to the community, now's a great time to say it, even though we're constantly in contact. I know there are lots of folks on here who don't have the pleasure of talking to you as regularly as Dom, Kat, and I do. So please, now's the time if there's anything you might want to say.
Well, I'd certainly say hang in there. It's, listening to all the presentations and the speakers, it's great just how many people are really invested in trying to get answers as quickly as possible. And all of the pieces, are attacking the problem from different directions. And I know families want therapies yesterday, but hang in there. We're doing our best. And I think together we'll be able to get there in the end and have, not just one, but, a suite of therapies that potentially can be applied to different situations.
That's a great point. We and our shoulder is behind you, and we're right next to you, and we know how hard you are working. So our thanks is beyond measure to both of you and to your entire team. Simran and John, thank you so very much for being here and for being here with us always.
Thank you. Thank
pleasure,
you, Luke.
Pleasure.
And I would really want to say thank you to each one of the KAND family. It really matters a lot. And what I want to say is it really motivates us every day in the lab, just thinking about each one of you in our heart and mind, and thinking that we're doing something in small steps, but eventually we'll be there. So thank you so much.
Thank you, John and Sim, for being here. And thank you to everybody for tuning in, and keep those questions coming. And we'll make sure that all these incredible researchers get those questions.